So tell us about the principal focus of your research.
So the principal focus of my research is to understand the pathogenesis of macular disease. This includes both inherited forms like recessive Stargardt's disease, but also age-related macular degeneration. And we address the events ongoing in retinal pigment epithelial cells. These cells and the underlying Bruch's membrane are considered by most people to be the site of at least the beginning of the disease process.
Now, the paper you presented here at AAO 2015 had to do with the origins of lipofuscin.
Right.
Tell us about the new interpretation that you've brought to this field.
The composition of RP lipofuscin has been a difficult thing to get a handle on for many years. I think it's now clear from the spectral properties of the compounds that we've isolated and characterized, and from their behaviors that the material is actually originates from the visual cycle and from compounds that form because of reactions of vitamin A aldehyde. And so these compounds are actually also the origin of fundus autofluorescence, which is used daily in ophthalmology clinics to diagnose and image disease processes. But these compounds are not just markers of the status of retina. They're actually part of the disease process. And this is clear in recessive Stargardt's disease, where it's clear that that's the accumulation of this material that kills the RP cells and leads to RP atrophy. We think that this is also probably part of the process in age-related macular degeneration also.
It's not a byproduct, it's actually an active participant in the process.
Yeah, so it's a byproduct to the extent that it's a product that has it doesn't have any purpose for the visual cycle. It's really an error in the constant cycling of vitamin A. These are aldehyde, so very reactive, and there are random events where the aldehyde is not being handled properly, and so it's allowed to react. These compounds that form they don't serve any purpose. They actually form initially in photoreceptor cells, and the photoreceptor cells have managed to take care of it by transferring it to the adjacent cells, and that where it accumulates, it gets deposited and accumulates over the lifetime of an individual.
Now, if you look at efflux mechanisms in this particular area, clearly there's a membrane transport problem. There's an efflux issue. Have you been able to quantify that in terms of lipofuscin removal?
Right. So there's often discussion about whether retinal pigment epithelial cells send the material out the back door, whether there's an efflux. There are some reports, histological reports of an autofluorescence basal to the retinal pigment epithelial cells. I think it's not really clear that this is happening. Certainly this material is not being undergoing lysosomal degradation as would all the other proteins and other molecules that are being phagocytosed by RPE cells. And we think that this is not a failure of the RP lysosomal enzymes themselves, but rather that they're being presented with something that they were not designed to degrade. And so these fluorophores just accumulate.
What is the role of complement in this process?
Well, I think that these are unusual compounds and have unusual structures. That certainly they undergo photodegradation, and we showed a few years ago that in concert with this photodegradative process, actually, they can activate complement. There may be a number of different things that activate complement in AMD, but these photodegradative products could be one of those factors.
So if you were to summarize the findings in this paper and tell us about the potential next steps for research in this area.
So it's not clear to us yet what the true level of accumulation is of these compounds over the lifetime of an individual, and what proportion actually undergo photodegradation. Obviously, there is less photodegraded than is formed because these compounds still accumulate. So that would be one area of interest to me, is to know how much of this is being generated, how much of the original compounds, and how much is being photodegraded. So how much of an insult is the eye experiencing due to the...
And you're using autofluorescence as a way to quantify this?
Yes, we use fundus autofluorescence, a quantitative method, yes. Unfortunately, that will tell us how much of these compounds are present at any given time. It doesn't really tell us how much may have formed initially and how much is photodegraded.
Are you doing longitudinal studies to try and delineate that?
Yes, we do longitudinal studies. We do quantitative autofluorescence longitudinal studies.
How many years have you accumulated?
At this point, actually, in our recessive Stargardt patients, we probably have about 3 years of longitudinal follow-up, and in AMD patients, not even that.
What do you think are the major triggers for this process?
So when we're talking about these visual cycle adducts, these are photosensitive compounds that are sitting, as they form, they're actually sitting in membrane, and they are light sensitive, so they can generate reactive forms of oxygen, and the lipids are sitting right next to them, so it's easy to imagine that they could be responsible for initiating the lipid peroxidation.
So you believe that that's the predominant initiator?
I don't have any evidence to suggest that it's the predominant initiator, but it certainly is positioned and has the correct properties to do so. But we just can't say how much.
It's a fascinating area of research.
When lipid peroxidation in photoreceptor cells, RPE cells, when it's discussed, it's the presence of light is discussed, the presence of high oxygen concentrations is discussed, but the actual photoreactive compound is not usually addressed.
Yeah. That may be a very, very important distinguishing feature.
Right.
And then, of course, the wavelength of the light. And,
the wavelength.
Intensity.
Right. Right. So these photoreactive events are not, they don't depend on UV light, for instance. These are compounds that are absorbing in the blue and even up to 500 nanometer excitation.
So we were to talk about the translational aspects at this point. If we believe that blue light is a part of the potential initiating complex, what are the clinical implications for that?
There are actually currently some clinical studies that are aimed at reducing the production of these visual cycle adducts, and they are addressing, of course, the need for therapies for recessive Stargardt's disease. But in addition, however, since the population of AMD patients is greater and also in need of therapies for the dry form, at least. A lot of the small molecule therapies that are under consideration right now would be applicable.
So turning to advice for patients, or putting this particular piece of research into perspective for an audience of patients or family members of patients, what are the implications of this particular line of research for them?
For recessive Stargardt's disease, since the accumulation of these compounds that is really responsible for the atrophy of RPE cells and photoreceptor cells, I think that therapies that could reduce the formation of this material are very important. They're really addressing the issue. Only better than a therapy like that would be gene therapy, which really cures the disease.
Is there anything else you'd like to add about your research, your program? How can people get involved in supporting your work?
At Columbia University, we actually have a very active recruitment of patients with recessive Stargardt's disease. The scientists that actually first described the gene responsible for recessive Stargardt's disease, the ABCA4 gene, is a colleague of mine at Columbia. And so we have a very active program and the largest cohort of recessive Stargardt disease patients. We also have a very large cohort of AMD patients.
So if there are family members or patients with recessive Stargardt's that want to get in touch with you, is there a website that they should go to?
Yes, they can through the Department of Ophthalmology at Columbia University Medical College. Through that website, they can reach individuals that will point them in the right direction.
Excellent.
Yeah.
Dr. Sparrow, thank you so much for joining us. We really appreciate this great interview.
Thank you very much.
Thank you.